Our first speaker is Carolyn Moores, who's going to be talking to us about molecular strategies for regulating microtubule dynamics. And this is going to be a bit on single particle and a bit of tomography. And then we'll have another tomography talk later.
Thank you. I'm going to stand here, I think. So thanks a lot to the organisers for the chance to present some of our data. We are interested in the biology of the microtubule cytoskeleton. Microtubules manifest themselves in lots of functional contexts. And what I'm showing here is the way in which microtubules are used as tracks for molecular motors. Maya talked a little bit about some of the work we've been doing with her on understanding the mechanism of kinesin motors, and I'm not going to talk about that at all today, but want to use that as a reminder to say how important our collaboration with Maya is on interpreting our cryo-EM structures. But what this picture of a cell, I hope, also emphasizes is the heterogeneity by which the microtubule cytoskeleton sort of manifests itself in terms of signaling for transport around a cell. There are lots of different types of heterogeneity that the microtubules use as part of that signaling process. And what I want to talk to you today about is microtubule-associated proteins, which are part of that signaling complex that molecular motors respond to. We're interested in that from a medical point of view, because, for example, in neurons and as part of neuronal transport, very many aspects of this cellular machinery can break down, and so we want to understand how this works in healthy cells and how this can then break down during disease. So I would like to tell you about two different microtubule-associated proteins that we've been working on. One, Doublecortin, is a long-standing favourite of mine. I even started working on that when I was a postdoc, and we've carried on working on it, and it's still continuing to provide us with some deeper understanding about how the microtubule cytoskeleton works. And second, I'd like to tell you about a much newer piece of work we've been doing on microtubule minus end binding proteins called CAMSAPs. I'm not going to say too much about the technical aspects of the cryo-EM and the structure determination we've been doing, but I'm of course very happy to talk about that. So this is just a little bit of a revision to take you back to your textbooks maybe. So microtubules are built from alpha-beta tubulin heterodimers. Alpha and beta tubulin are very similar to each other. And they are both nucleotide binding proteins, so they both bind to GTP. But the position of beta tubulin in the context of the dimer means that it's exposed on the one of the surfaces of the dimer, and that has a profound influence on the way in which the microtubules are put together and the way in which their dynamics are regulated. So if we draw a schematic of the tubulin dimer like this, there are multiple surfaces by which these tubulin subunits can come together to form different kinds of oligomers. But in the context of the microtubule, the first organizing principle of the dimers to consider is the longitudinal contacts at either end of the somewhat elongated subunits. And these form head-to-tail contacts between adjacent subunits and form a protofilament, and the polar organization of these subunits means that the protofilament itself has a polarity. So where the beta tubulin is exposed, it's called the so-called plus end. The tubulin subunits tend to add faster at that end, and that's why it's called plus end. And the minus end has a slightly slower type of dynamics, but it contributes to what's going on in the cell, and I'll talk a bit more about that later. But alpha tubulin is located at the microtubule and protofilament, so-called minus end. And then what's also very important about these longitudinal contacts is that the GTPase site of beta tubulin is found at these longitudinal contacts. So it's informing these protofilament contacts between one alpha tubulin here and the beta tubulin here that actually activates the enzyme of the tubulin. And so the enzyme activity of the tubulin is inextricably linked with the polymerization properties, and this is the way in which microtubule dynamics are driven. So once you've formed a protofilament, then these protofilaments can then form lateral contacts side by side with neighbouring subunits. Typically, you will form a contact between a beta-tubulin and a beta-tubulin, so so-called homotypic contact. I'll come back to the exception in microtubule structure where that's not true. But in building up protofilaments longitudinally and laterally, you will finally come to form the cylindrical structure of the microtubule, and it's then in the context of that cylinder that one needs to consider the enzyme activity and how the overall polymer is put together. So this is also summarizing of course, a lot of biochemistry data. So in the presence of GTP, nucleation and polymerization of tubulin is very strongly stimulated. As we just discussed, in forming these longitudinal contacts, the GTP hydrolysis of tubulin is then stimulated such that most microtubules will actually be built of GDP tubulin, because the hydrolysis will have caused formation first of GDPPI and then release of the phosphate. So GDP tubulin is quite unstable in the context of the dimer. So as long as addition of GTP tubulin is staying ahead of hydrolysis, then microtubule will continue to grow. But once hydrolysis catches up with polymerization, then you're left with a microtubule that's built mainly of GDP, and this is very unstable and it falls apart, and then this cycle continues. This process will be, in the presence of sufficient GTP, this process will happen continually in solutions of pure tubulin in a stochastic fashion. But of course, in the context of cells where there's very precise requirements for microtubules to grow and shrink, leaving microtubule polymerization to stochastic processes is not very helpful. And so there are numerous microtubule-associated proteins that control various aspects, in fact, nearly all aspects of these different stages in microtubule dynamic instability according to the requirements of the cell. So that leads to some ideas about how we would like to understand the roles of these different microtubule-associated proteins in controlling microtubules. But also, by studying individual maps, we can also shed some light on how dynamic instability is driven. And what you can also think about by looking at this already extremely schematic, simple schematic, is that there's actually quite a lot of heterogeneity biochemically, and presumably also structural heterogeneity, and we would like to use manipulation of the system in a variety of different ways to understand that. So of course we've got the biochemical heterogeneity between GDP and GTP tubulin, for example, and of course you've got the heterogeneity which is occurring at the microtubule ends, and it's actually at the ends where the microtubule dynamics are actually regulated. So I'm showing this schematic shows dynamics only occurring at a single end, but as I said, both plus and minus ends of microtubule dynamics. Microtubules are dynamic, and we would like to understand those areas too. So cryo-EM has been a very powerful methodology for understanding microtubules' structure and function for a very long time. And of course, they're very beautiful in vitreous eyes. So this is actually a kinesin-decorated microtubule. And what you can see here is that the microtubule itself has a diameter typically of about 25 nanometers. And these individual little bobbles along the edge of the projection of the microtubule are individual kinesin motor domains. These are about 40 kilodaltons in size, but because they're regularly arranged along the microtubule lattice, you can see them organized very clearly. In fact, in most of our single particle type studies, we use the regularity of the microtubule lattice to illuminate, first of all, how the microtubule is organized, but also how the binding proteins interact with that regularity, and that provides us with sort of pseudo-crystalline array of the microtubule regulators that we're interested in. So as I said, we're typically working with a sort of single particle approach to microtubules, essentially breaking them up into small chunks and then reconstructing them in those chunks. We're typically working with purified tubulin stabilized by some means or another, and then adding proteins by molecular reconstitution to them. But recently we've also ventured into using electron tomography to understand the more unique structures which are formed by microtubules, for example, at their ends. So I'm going to be mainly talking about these sort of single particle approaches, but also showing a little bit of electron tomography data too. First of all, I'd like to tell you a little bit about the background of why we're interested in working on the microtubule stabilizing protein called Doublecortin. It was originally identified because when it's mutated in humans, there's a very dramatic brain developmental disorder that is caused by that. So you can see here the normal convolutions of a healthy human brain, and here you see a very so-called smooth brain of a person who has mutations in the Doublecortin gene. And so functionally, Doublecortin is obviously very important for brain development, and specifically it's important for regulating neuronal migration during brain development, of which there is a great deal. But members of the Doublecortin family of microtubule stabilizing proteins have been identified beyond mammals and have roles beyond regulation of microtubule dynamics in migrating neurons. And there are homologs identified, for example, in both worms and in flies, and they have different roles in regulating microtubules and different functional contexts in those organisms. In terms of what the protein actually looks like, one of the very striking observations about the original characterization of Doublecortin as a regulatory protein in neurons is that actually neurons are really full of different types of microtubule regulatory proteins. So it was already gave us an idea that some of the properties of Doublecortin must be rather unusual compared to other microtubule stabilizing proteins, because none of them can compensate for the lack of functionality in these patients. So one first unusual observation with Doublecortin is that actually it has some domain structure, which is rather unusual compared to, for example, MAP2 and tau, which are more classical neuronal maps. And in particular, these repeats of these so-called DC domains have been shown to be very important in binding to microtubules, and in particular, lissencephaly causing point mutations cluster within these two domains. So this converges to a summary of what we think Doublecortin is doing with respect to regulating microtubules. This is the structure of a DC domain. Those two DC domains bind to microtubules and regulate them in some way. We know that the GTP activity of the GTPase activity of microtubules is retained in Doublecortin stabilized microtubules, but Doublecortin is particularly good at nucleating microtubules, and in particular it's very good at nucleating microtubules built from 13 protofilaments, which is, in the case of eukaryotic cells, a lucky number, because 13 protofilament microtubules are the predominant architecture in nearly all of the cells in our body. So Doublecortin has evolved specifically to stabilize that particular architecture of microtubules. So we, of course, wanted to know how does that work? So our experiment in trying to understand Doublecortin's interaction with microtubules is to begin with tubulin itself. These microtubules will be rather dynamic because of the GTPase activity, and so we can stabilize them by copolymerizing with microtubules. And because we're copolymerizing with Doublecortin, we're going to get a largely predominant architecture of 13 protofilaments in this sample. And at the time when we started doing these experiments, we didn't know where Doublecortin was binding, and so we wanted to add a fiducial marker to the microtubule to try and help us localize ourselves with respect to the rest of the structure. So we used, in fact, a kinesin motor domain, the binding site of which is very well understood, to then tell us where Doublecortin was on the microtubule lattice, having validated that that didn't seem to affect the behavior of either Doublecortin in the presence of kinesin or vice versa. So this was our original sample that we use for structure determination, and this is now seeming to be quite a historic or artifactual artifact is the structure of the 13 protofilament stabilized by microtubule stabilized by Doublecortin in the presence of kinesin. And let me just introduce you to the structure itself, which is the wall of the microtubule is in blue. The kinesin motor domain, this 40 kilodalton quite well-defined structure, is in red, and that means that the unaccounted for density corresponds to Doublecortin. And what this reconstruction was able to tell us is that Doublecortin binds at the corner of 4 tubulin dimers, which is a great place for a microtubule stabilizing protein to be binding, because it's able to, by binding there, it stabilizes both lateral and longitudinal contacts within the microtubule lattice. If you can see a little bit from maybe the side views, you can also see that Doublecortin sort of binds in between, in the valley in between the protofilaments, and that gave us a clue of how Doublecortin is probably acting to favor the 13 protofilament architecture from which the microtubules are built. Because you can imagine that by binding in between the protofilaments, there's some sensitivity to the angle between the protofilaments as they form the hollow cylindrical wall of the microtubule, and therefore ultimately has an influence on how many protofilaments can actually fit around the cylindrical wall of the microtubule. So this view is the original single particle reconstruction of that data, but we, by considering each of these protofilaments as essentially identical, we could then apply some additional averaging around the wall of the microtubule. Oh, and what I should say before I go on to that point is that in 13 protofilament microtubules, there is a discontinuity where instead of having beta tubulin next to beta tubulin, you have alpha next door to beta. And what we were able to see in our reconstruction is that Doublecortin can tell the difference between these binding sites. So here, it's contacting an alpha tubulin here, and a beta tubulin here and here. But at this intersection here, where you can see that the kinesins don't match up so well, there will be an alpha tubulin next door to a beta tubulin, and Doublecortin doesn't bind there. So the binding site of Doublecortin is very precisely defined by the tubulin subunits with which it interacts. So if you then use the symmetry around the wall of the microtubule, you can get a much closer look at what Doublecortin is looking like and the way in which it's contacting the 4 tubulin subunits formed by its binding site. And in fact, it makes a very precise set of interactions between these 2 alpha tubulins and these 2 beta tubulins. It's these points within the otherwise relatively well-conserved sequences of tubulin where there are differences between alpha and beta tubulin and to which Doublecortin is apparently sensitive. Doublecortin sits on the outer surface of the microtubule, but in fact the stabilization that maintains the cylindrical structure of the polymer is formed by lateral contacts on the inside. Doublecortin doesn't reach through to the inside of the microtubule cylinder, and so it's the idea by which Doublecortin stabilizes the microtubule is to facilitate the proximity of the tubulin subunits, and then allows them to form these lateral contacts between the alphas and betas across the protofilaments, and this allows stabilization of the microtubule cylinder. These structures are not specific to Doublecortin stabilized microtubules. These are very typical of microtubules stabilized by any number of different reagents. But what was very interesting for us when we first saw this structure was also these are microtubules that are stabilized only by Doublecortin and not, for example, in the presence of the stabilizing drug Taxol. But we can see that the pocket where Taxol would bind actually exists in this structure. So this is a pocket that it has evolved to be formed in the context of the polymer, and Taxol is able to capitalize on this somehow and to stabilize these microtubules by that means. But Doublecortin doesn't perturb that pocket in the context of these microtubules. So that was then, and of course we are continuing to work on that on this question at higher resolution. So this is what Doublecortin stabilised microtubules look like now. This is data collected on our Polara at Birkbeck. And there are a number of questions that we can now begin to address much more comprehensively with this kind of data. One of the questions relates to how the Doublecortin interaction with the microtubule lattice responds to the stabilization in the presence of different nucleotides, for example. And so we know that the Doublecortin binding site is very close to the GTPase active site of beta-tubulin. And so one of the things that we can now start to look at is what's actually going on in terms of the nucleotide occupancy of that catalytic site. And what's handy about tubulin is that there's a sort of built-in control because alpha-tubulin, as I said, also binds to GTP, but it's never hydrolyzed in the context of the lattice. So this is the GTP that's fitted into the structure of Doublecortin stabilised microtubules in alpha-tubulin, and this is the GDP, which is present in the beta-tubulin active site in the presence of Doublecortin. So we knew biochemically that Doublecortin supported hydrolysis of GTP, but now we can see that directly by looking at the nucleotide, and this having the power to see this kind of detail in the active site also allows us to address questions in terms of sensitivity in the presence of different nucleotide analogues, and that's yielding some quite interesting results, both in terms of how Doublecortin responds to the biochemistry of tubulin, but also what it can tell us about how tubulin itself is responding to the different nucleotides. So the other question that we've been struggling with for quite some time, and which you will probably have spotted in the course of looking at the structures that I've already shown you, is that we don't, the density that we see for Doublecortin on the microtubule lattice corresponds only to a single DC domain. You'll see that the resolution that we have in our reconstructions is quite a bit worse in the presence quite a bit worse in the Doublecortin region of the reconstruction compared to the tubulin portion of the reconstruction. And so that there could be a number of explanations for that, but we think we know that at least one of the things that's contributing to lower resolution in this region is heterogeneity of occupancy of the different domains within Doublecortin, because in principle you could think that this domain is occupied by maybe exclusively NDC, and that the rest of the protein is completely flexible and not seen at all in our reconstructions. Or you could guess that it's actually a combination of the two, possibly from within the same molecule, because certainly this 40 amino acid linker is long enough to stretch across the different possible binding sites for the different DC domains. We actually don't know the answer to that particular mechanistic question, but we can certainly see, if we compare structures of NDC, shown here in green, docked into this DC density, compared to CDC, docked here shown here in purple, that although there are regions of very good match between the DC domain and our density, there's this loop region which is very different between NDC and CDC, and actually neither fits well in the density that we can now visualize, which suggests to us that actually we have a combination of both of the domains binding on the microtubule lattice. And this leads us to think about different ways of testing this idea. One of the things that we are currently doing is some DC domain engineering, so making chimeras of Doublecortin with either two NDCs or two CDCs, because that will allow us to address both functional and structural questions about what these DC domains look like on when bound to microtubules. So this is our areas of interest for studying Doublecortin, and then more recently, we've been working with the group of Anna Ackmanova in Utrecht, who some of you will certainly know, to study these interesting microtubule minus end recognizing proteins called CAMSABs. So for a long time, actually, the minus end of microtubules was thought to be completely boring and inactive. This is in large part because in many cells, the minus end is capped by the gamma-tubulin ring complex, and actually there are no dynamics occurring there because the gamma-tubulin ring complex binding prevents it. But in a number of cells, increasingly, but the identification of this family of proteins, with this not totally memorable name, is showing that the minus ends of microtubules are actually very important physiologically. So there's a number of examples and a variety of metazoa. This is a nice image of epithelial cells in which, in contrast to many sort of more classical views of microtubules in cultured cells that have a radial array, in epithelial cells the microtubules are organized top to bottom in the cells, and actually you can probably just about make out here, and it's easier to see in this view, that the minus ends of microtubules are organized very precisely with respect to the tops of these cells, and that's very important for the physiology and functionality of these cells as they operate in the context of the epithelial layer. So there are a lot of different physiological contexts in which these proteins are now known to be important, but it has a very this raises some quite intriguing questions about what is the specific recognition mechanism by CAMSAPs for these microtubule minus ends. And beyond their physiological context, they are known to be important in these different, more precise functional roles, and in particular, their role in protecting the minus ends of CAMSAP-decorated microtubules from the actions of microtubule depolymerizing enzymes is quite well characterized. So we'd like to understand how they work. So CAMSAPs are complicated multi-domain proteins, large multi-domain proteins, but as they were increasingly characterized, it became apparent that their so-called CKK domains, also not rather sort of prosaically named, are very important mediators of the microtubule minus end recognition, and so we thought that this would be a good structural target to try and understand a bit more about how in the full-length protein context, how microtubule minus ends are recognized. What our collaborators in Anna Akmanova's lab were first able to show is that the CKK domains themselves actually have some capacity to recognize microtubule minus ends. So it's not, it's by no means as total a recognition as the full-length proteins, but the CKK domains have some intrinsic microtubule minus end recognition properties. And that's actually seen in both CAMSAP 1, 2, and 3, which in the context of the full-length proteins have quite different properties with respect to microtubule minus end recognition. So the CKK domains really did seem to be a good target for trying to dissect this property of the proteins. What you can also see, though, is that the CKK domains bind also to the microtubule lattice, and that gives us a little bit of a route into understanding their structure in terms of more classical single particle studies of microtubules, and from that we hope to sort of extrapolate how mechanistically the minus end might be recognized too. This was the context for our structural work. There was an NMR structure of a CKK domain deposited by a structural genomics project some time ago, but it was essentially a domain of unknown function at that point of deposition. So our collaborators, Michelle Steinmetz, who many of you will know, resolved the structure just so that we could be very clear about the set of interpretations we were going to be making about this domain. And this is what it looks like. It's a relatively small and compact domain with several rather flexible loops that are intriguing in terms of interactions potentially with the microtubule. So our experiment was to take the CKK domain, actually from CAMSAT1 and CAMSAT3, and solve their structures using first by single particle cryo-EM. And this was the initial output. So this is the asymmetric reconstruction from a single particle experiment. So what you can see straight away is that the density that corresponds to the CKK domain is binding every 8 nanometers on the microtubule lattice. And once again, it does not bind to the microtubule seam that's very characteristic of the 13 protofilament microtubules that we were using for our structural experiments. Using the same averaging technique to average multiple protofilaments together, we were able then to focus in on the organization of the CKK binding site on the microtubule lattice. And this allowed us to see quite precisely the way in which the CKK structure binds to the microtubule, and in particular the location of its binding site on the microtubule lattice, which is actually at the in-between two tubulin dimers. So here's an alpha tubulin, here's a beta tubulin, and here's the second pair. And what you would see if you turn this reconstruction around is that these are taxol stabilized microtubules, and in the tubulin density, this is certainly good enough to identify where the taxol is binding, and therefore that this is a beta tubulin, this is an alpha tubulin. What you will probably be spotting is that although the overall resolution of our reconstruction was about 5 angstroms or so, actually the density corresponding to the CKK domain was a bit worse than that. We typically see that our overall resolution is dominated by the tubulin density, which is always better, nearly always better than our binding protein. Nevertheless, we can see very clearly the way in which the CKK domain docks within this density, and this already tells us some interesting things about what we know about the biology and functionality of CAMSATs. First, you'll remember I said that the CAMSAT binding to microtubule ends protects them from depolymerization by kinesins. And we know that the kinesin binding site is here. So if you have a CAMSAT bound to a microtubule, the chances are that there won't be room for a kinesin to bind to it too. And so probably CAMSATs can compete with kinesin 13s at microtubule minus ends by steric competition for overlapping binding sites on the microtubule. What you will also hopefully remember is that this is a binding site that's 4 nanometers away from where I showed you Doublecortin is binding with the microtubule. So there can be there are different implications from that alternative binding site. As you'll remember, I mentioned that by Doublecortin, by binding here, expresses some can express some sensitivity to the nucleotide. The state of the microtubule with which it's interacting, and so-called M-binding proteins also bind at this point and are extremely sensitive to the nucleotide sites of the nucleotide state of the microtubule. So by binding here, CKK probably is not sensitive at all to the nucleotide binding content of the microtubule, and there are several functional reasons why that would be important. So the interaction between the CKK domain and the tubulin surface in the microtubule is spread across the entire CKK domain. And again, there are very precise contacts formed between, in particular, many of the flexible loops within the CKK domain and the surface of the microtubule. What we then, of course, wanted to do was to be sure that, or to investigate the ways in which those interacting regions were affecting the interaction of the protein with the microtubule lattice, and then start to reformulate our ideas about our structure on the lattice in terms of thinking about end-binding protein, because we would like to know that the structure we've solved here has some relevance for the interaction with the microtubule ends. And so Kai, the postdoc in Anna's lab, basically mutated everything at the surface, the interface between the CKK domain and and the microtubule. And you can see that in many cases, mutation of, for example, charged residues tends to result in a slightly lower affinity for the microtubule end compared to the wild type. And this is what you expect typically for residues that are affecting the affinity of both of the protein for both for the lattice and for the microtubule end. But you'll probably also be spotting that there was one very anomalously behaving mutant, which is this one here. So you can characterize the effect of the mutants in different ways. But this mutant in particular seemed to yield a completely opposite effect, which is to really enhance the affinity of the CKK domain for the microtubule lattice. This was not what we were expecting at all, but we thought that by studying the structure of this mutant bound to microtubules, that might yield some insights about the way in which the CKK domain can differentiate between the microtubule lattice and the microtubule end. So the structure of this mutant was solved in the same way as the wild-type structure had been solved. It binds at the same place in between two tubulin dimers. And what's very interesting is that there's only very subtle differences between the structure of the wild type and its interaction with the microtubule and that of the mutant. So the wild type is shown in green here and the mutant is shown here. This residue is exactly at this interface here and will be directed towards the tubulin in its binding site. So it's not completely trivial to actually interpret that result, in fact. But what we could certainly conclude is that this mutant is not having an effect on the fold of the CKK domain, and in fact, overall, the binding site is preserved in the context of the mutant. But in displaying that very profound effect in terms of interaction with the microtubule lattice, there's only a very small structural effect in terms of the behavior of this protein with its microtubule substrate. And so we concluded that probably, therefore, the mechanism by which the CKK domain is recognizing the microtubule end compared to minus end compared to the microtubule lattice is also rather indicative of quite a subtle conformational difference between those two tubulin organizations. So it then becomes a quite sort of a philosophical question: what actually is a microtubule end? Because there can be lots of factors that contribute to endness. So one of the things one of the mechanisms by which ends are quite well defined in dynamic microtubules is the presence of GTP as opposed to GDP in the majority of the body of the microtubule. And indeed, end-binding proteins are quite well established now to recognize different nucleotide states within the Tubulin GTPase cycle. And that by that means they dynamically interact with the growing ends of microtubules. In contrast, kinesin 13s, which, as I mentioned, they're depolymerizing microtubules, they are thought to recognize some kind of non-lattice endness about tubulin, which is probably some kind of reflection of the flexibility of tubulin that's not held completely in the constraints of the microtubule lattice. Whereas gamma-tubulin ring complexes, which are classically thought of as protecting microtubule minus ends from microtubule dynamics, they recognize and bind to the end surface of alpha tubulin at microtubule minus ends. So there are these sorts of mechanisms which we conceived of as that could be ways in which the CKK domain recognizes the microtubule minus end, and we tried to dissect which one was the best. So we could establish quite clearly, in fact, that regardless of how the microtubules are stabilized, the CKK domain will always bind to the minus ends. In addition, the fact that its binding site is not located adjacent to the GTPase site of beta tubulin, which is also a site of structural rearrangements during the course of the Tubulin GTPase cycle, supported the idea that CKKs are not recognizing anything to do with the nucleotide state of the microtubule minus ends. In terms of recognizing the end surface, the binding site of the CKK domain that we could visualize suggested to us that it's unlikely that there was any sort of recognition component about the end surface of alpha tubulin at microtubule minus ends. And the only very subtle differences that we saw between the wild type and the mutant also sort of suggested that's slightly less compelling in terms of evidence, but we were satisfied that we could probably eliminate that as a mechanism. So that left us with something non-lattice, like about microtubule minus ends in particular, compared to the lattice and also compared to the microtubule plus ends. But so one of the other things we were able to, that Anna and Kai were able to establish is the location of the peak of CKK binding at microtubule minus ends with respect to tubulin. So if you use statistics on multiple TERF images of decorated microtubules, one can gain a very precise idea of where the protein is binding with respect to microtubule ends. So if you notice here, the scale is quite large here, but what they're studying is the peak of binding fluorescence from the CAMSAT protein with respect to the microtubule. If one focuses in more precisely here on the microtubule end, which is designated by the end of the tubulin signal in this data, although the spread of signal from CAMSAT protein is quite broad, there's a peak of binding activity which is not exactly at the microtubule end. So there's something a little bit behind the microtubule end that the CAMSAP is tending to bind to and recognize. And so there's some interest then in trying to understand what the structure of that actually is. And there's so this was the point at which we turned to electron tomography to actually study the structures of non-dynamic microtubules to which we know the CKKs and CAMSAPs can bind. So this is a relatively simple experiment compared to some of the other tomography experiments we've been hearing about, but we would like to understand what the microtubule end structure actually is. So we collected tilt series of non-dynamic microtubules and looked at their ends, and because of the structure of the microtubules we can tell which is a plus end and which is a minus end, even though we can't see the very precise underlying structure of alpha and beta tubulin, for example. And we can see very clearly that the ends of microtubules don't stop abruptly. They actually flare out in a quite gentle way, forming two-dimensional sheets that are curved both longitudinally and laterally. We did some quantitation of these ends, and actually in terms of the longitudinal curvature, which is the way in which the sheet curves away from the axis of the microtubule, there's no real difference between the plus and the minus ends. So longitudinal curvature by itself is not can't be an explanation for the way in which the CKK domains are recognizing minus ends. What we did notice, however, compared to the curvature of tubulins that are often observed in tubulin PDB structures, these ends are very gently curved compared to those. So we can see, we could also see that each end is actually unique. So there's something common, there must be something common in between all these ends, which is not dependent on the very precise organization of the protofilaments in each case, for example. Certainly these protofilaments are close together compared to the classical ram's horns type view of microtubules that are depolymerizing. So this is now slices through the microtubule to show you a different type of curvature that we're seeing, which is that as the protofilaments start to stop, as you start to approach the microtubule end, the lateral contacts that hold the cylindrical wall together start to relax, but the contacts are retained, so a sheet is formed as opposed to a hollow cylinder, and so that says that the protofilaments move both away from the axis of the microtubule cylinder, but are retaining the connection between them. What is interesting about the retention of lateral contacts in between the protofilaments is that that means that in principle the CKK binding site that we observed which is also in between protofilaments, is preserved in these sheets. And so by this observation, we conclude that in these sheets that binding site is preserved and presumably optimized actually in the context of the microtubule minus end. We, from this set of data, we have concluded that we have an idea about what's going on in terms of recognition of the microtubule minus end. And it comes from a combination of both longitudinal curvature and lateral curvature, which is rather specific to the microtubule minus end, but rather subtly defined compared to both the lattice and the plus end. So you can imagine in principle that there are 3 different zones on a microtubule, and they're defined by 2 different types of curvature. So we've already discussed extensively the longitudinal curvature on the lattice, of which there is none, and the lateral curvature, which is defined by the protofilaments from which the microtubule is built. And at the both plus and minus ends, that longitudinal curvature starts to come into place, and we, as I described to you, we didn't see differences between the plus and minus ends in terms of longitudinal curvature alone. However, if you imagine now how the lateral curvature changes as you transition from lattice to end, and how the lateral curvature at the minus end will be more pronounced in the alpha tubulin compared to the beta tubulin, which is a different situation than how it is on beta tubulin compared to alpha tubulin at the plus end. And so by this mechanism, we think that the CKK domain is fine to bind to the lattice binding site, is really not able to bind to this very flattened region of beta tubulin, but binds very comfortably in the alternative curvature of the microtubule minus end. And so by this means, we think the CKK domain can recognize this curvature and therefore recognize microtubule minus ends. We, of course, have work to do in terms of putting this behavior in the context of the full-length protein, and very many other questions to address about the effect of microtubules on this kind of binding. And obviously we need to understand what the role of the rest of the protein is in this activity. So this is where I would like to stop. We think by studying these 2 proteins we've understood some interesting things about them and also about the microtubules to which they bind. I should absolutely make sure to acknowledge that the work I've showed you has been done mainly by 2 postdocs, one of whom is here. So Shimon has been continuing the work of Frank on Doublecortin at high resolution, and Joe has done all of the work on the CAMSAT proteins. He's here. He can tell you more about that project and the way we want to take that in the future. And I'll stop there. Thanks a lot.
Thanks. Thank you, Carolyn. That was really interesting. We have seemed to have run out of time for questions now.